Hello, everyone. Welcome to Foresight's Neurotech group. There probably are a couple people that are joining us from the Luma calendar. And for those of you that are uninitiated to the Neurotech group, this is a meeting run by Foresight to highlight current neurotech research. If you're interested in potentially being a part of it, if it's up your alley, you can send me an email at lydia@foresight.org, and I'd be happy to onboard you. Also, in other news, housekeeping news, we're hosting a neurotech research workshop where we'll focus on whole brain emulation and BCI development. And this will happen in October. And so I will post the link in the chat if you're interested. And as always, you can always ask me some questions through email. So without further ado, today we have Akash Kulgod with us from Dognosis, a deep tech startup focused on digitizing scent perception using dogs, machine learning, and BCIs. He's a Foresight Fellow in the 2025 Neuro cohort, and his background is in CogSci, and he's done research at 8 labs around the world. So thank you, Akash, for joining us today. We're so excited to learn about how one would teach AI to use machine learning to track the scent of cancer. So please take it away.
Yeah, thanks so much, Lydia. And it's really nice to be able to make one of these. I've always been meaning to, and I guess I had to be speaking at one of them to finally make it. But really glad and also really excited for the BCI workshop that I'm hoping to make in October. Cool. So I'll be going on a bit of a walk. I have 3 stops. The first is just a short primer about Dognosis, what we're doing, what we've done so far. And then I want to dwell a bit on the big idea behind Dognosis, and I think it's especially relevant for this group because it focuses on how neurotechnology and other things that we're learning about the brain can actually unlock a new sense. And then I'd like to end with what we've been doing this year specifically and what we have planned. So that's the map of the walk. So the main agenda, the main focus for us at Dognosis is the following problem statement. In India, 1 million Indians could be saved every year if we just detected cancer early. So when you detect cancer early in stage 1 and 2, generally when it's asymptomatic, it hasn't spread, you have extremely high success odds for treatment, those orange bars. And if you detect it late, it can often just be 6-12 months too late. You just plummet to those low green bars. And in India, this happens in 80% of cases, but it's not just an India problem. It's a global problem. So cancer screening remains unsolved. This is for many reasons. It's quite expensive to screen for cancer currently. It's often uncomfortable, depending on these invasive, almost medieval-like procedures. It needs to happen for every organ, for the few organs that we have a screening method for, which isn't that many. It's about 4 or 5, depending on who you ask. And then it needs to happen periodically. And given the fact that this needs to happen for an otherwise healthy population, it doesn't happen at all. So in India, less than 1% of Indians are screened in their lifetime for cancer. The numbers can be better depending on which country you're in, but the problem remains. And it's a public health problem, which means it's a pretty hard problem. It's also a Bayesian problem. It doesn't really matter how good your test is, because in some sense, you're fighting Bayes' law, where the prior of having the disease dictates the outcome quite strongly. And ultimately, we think it's a technology problem because the former 2 problems can be helped a lot if you just had better technology, which is something that a lot of people are now working on, but it remains really hard. So Siddhartha Mukherjee has a great piece about a month back about just the challenges in detecting cancer early, even with advances in genomic biomarkers, liquid biopsies, but there's still plenty of catches, a lot relating to this Bayes' law problem. So at Dognosis, we think the solution has been lying right under our noses. At this point, I should probably pause and apologize for the puns that are yet to come. But we know that breath carries a cocktail of volatile organic compounds, VOCs for short. Breath is far richer in these VOCs than urine or blood, contains almost 1,500 of these compounds. And what we know with 30 years of research is many diseases carry a scent signature that is consistent across individuals. So their breath has the same signature for the same disease. And what we also know is with around the same number of decades, we know that with a sophisticated enough sensor, with enough compute, and a clever enough algorithm, we can decode the scent pattern, and we can do so quite accurately. And that's just what dogs do. So, for about the last 2 decades, there have been multiple peer reviewed studies. It's been pioneered by this group in the UK called the Medical Detection Dogs. They did the first study back in 2004, but many groups all over the world have replicated this. And that's what we're building off at Dognosis in our lab in Bangalore. And the really interesting piece about the dogs is it turns out they're really good. So when you give them the right data set and the right conditions, they can often hit or even surpass some of our best diagnostics. So this is a selection of studies. It's from this great review paper in 2021 that covered about 50. They were double-blind studies, a variety of cancers, even diseases like bovine viral diarrhea. Of course, COVID-19 was really the watershed moment because multiple groups, about 60 all across the world, were able to train dogs to detect COVID. In one of the studies that I highlight here by Hag Ali, the dogs are better than gold standard PCR, which was being used to assess them. And it turned out they were better actually than the yardstick that was being used to assess them. And there are many more such cases. So it's interestingly enough, a Guinness World Record exists for the most diseases detected by a dog group. It's held by medical detection dogs. The number is 28. What we're doing at Dognosis is saying, okay, we have one of the best olfactory sense makers on the planet. It's a dog's nose and a highly trained detection dog at that. And what we can do is fuse their intelligence with neuro AI that is trained on brain computer interfaces, sensors, as well as vision data. And that can create the best in class platform for detection from breath at scale. And a snippet of what this looks like is in this video, which is from a few months ago from our lab in Bangalore. We have one of our dogs. In here, it's Iggy. She's sniffing a face mask in each one of those 8 ports. And while she's doing that, we have a bunch of sensors in the platform, cameras, as well as EEG, wearable BCI, and some other sensors like IMUs, record from all. And she still is making the decisions. She's saying whether she thinks a sample has cancer or not. And we know which samples have cancer in this setup. So we're collecting it from people who've already been diagnosed or healthy controls. And as Iggy is making the decisions, we're tracking her decision making through the multimodal data streams that we're capturing. And the goal then is to train machine learning models to tell us, instead of just a yes or no that Iggy could give and a handler, such as my co-founder, could assess whether she's making that decision, we can now do this with the machine learning models that we train. And suddenly we have a multimodal, multi-dimensional signature instead of a binary yes/no. And that's the idea that powers our main product that we're working on, which isn't really in neurotech, but it's powered by neurotech. So it's a multi-cancer early detection test where individuals breathe into a mask. The mask is sealed. It's shipped back over to a lab where a team of dogs and sensors can then provide a highly predictive risk score that can be used by clinicians to then guide whether the person needs a follow-up test or even treatment. And what we've been doing over the last year and a half is run this test that we're calling Breathe Easy in a phase 2 assessor-blinded study. So the sample, like I said, is a mask that people wear for 10 minutes. It's a regular mask. It costs about 15, 20 pence, half a dollar max. We've collected over 6,000 unique samples, 4,500 of which went into a training set in the development of both the dogs as well as the models. And then we used 1,500 in a test set. And we worked with a great set of hospitals across Karnataka in South India to do this. And what we found was this was our first data set that we presented just a few months ago from last year, where in a novel test set of 200 samples, the dogs achieved a 96% sensitivity, which meant that if someone had cancer and they had given their mask, the dogs got it 96% of the time. And they achieved this with just 10 weeks of training. So it didn't take that many days or samples for the dogs to get really good. And this is actually just from today. We just completed an analysis from our new test set with 1,500 samples. Again, assessor mask, basically replicating the past data set with a much larger set of samples. And what we were able to find was the accuracy and sensitivity remained above 90%. And that sensitivity was consistent across early and late stages, which is, of course, crucial for early detection. And this is fairly unprecedented. The best liquid biopsies achieve a sensitivity of around 17 to 20%. And it's just a testament really to what the dogs can do with their noses that current genomic markers and other advanced technologies can't. So we were pretty happy that we are able to reproduce this result in a much larger and novel test set. And what we think this ultimately would entail is a scalable model to do that first layer of population screening and tackle that piece of the Bayes law problem with a team of 20 dogs in one of our labs or our current lab. We can do 1 million tests a year with the dogs just sniffing for about half an hour or so because they're just really quick. So the dogs get to play the game of find the target scent for 30 minutes, which they also really enjoy because it's cognitively enriching for them to do scent work. And then we can screen 1 million people in a year. And what's I think really cool about this is if you do the math of the sensitivity that we have, we think a single dog at diagnosis can save around 100 lives every year. So we've been presenting our work, more papers and presentations to come. We've been happy to get some really nice organic press features as well in different avenues. We have a great set of collaborators, including hospitals in India and 2 of the leading canine science groups in the world. And we have an amazing team. So this is the whole team. It's now 50 plus strong. We were also amazed that we reached this level, but it was really cool. We did a retreat last month in our lab/farm. So this is all the dogs, all 12 of them, and about 40 humans. And yeah, it's been really great working with everyone on this project. So now I just want to talk a bit more about the big idea behind diagnosis and what I like to call sniffing the olfactory code, a bit of a play on what usually people call tracking the olfactory code, which is one of the unsolved areas of neuroscience and cognition. And in some sense, it's the holy grail of the field of machine olfaction. And it starts with this observation that in 2025 now, GPT-5 just released, we have AI that has arguably human comparable senses of vision and hearing, as well as language, reasoning, and other cognitive abilities. But what about the sense of smell? And my Berkeley thesis advisor had a great line about how humans are blinded by vision, which I think really speaks to how, as just visually oriented cognitive agents, we don't really think about this whole world of smell. And that isn't completely true, right? So people have been working on digitizing the scent of smell because we know how much that could unlock. So DARPA, as well as labs across the world, have invested billions in building what is called an electronic nose or an e-nose that can rival a real nose and especially a canine nose. But still today, governments around the world are relying on dogs at borders, airports, and the battlefield. And one of the fundamental reasons is the sensor itself. So the dog's nose is just seeing at a resolution that our sensors still cannot match, especially for multiple compounds. So the dog is seeing in parts per trillion. Our most advanced analytical instrument sees in parts per million. That's gas chromatography, mass spectrometer. That's a picture on the right is an Orchid's IDF dog detecting a bomb. It's one of the reasons why we still rely on dogs. It's this ability to really see at a much higher resolution. But it's not just the sensing. So the sense making as well, and I think this is captured really well in this 2021 paper by Andreas Merschen and colleagues, who's a good friend of Diagnosis. And he has a line in this study where they were looking at integrating canine olfaction with GCMS about how the scent signature of cancer is encoded in the brain, not by a particular set of molecules, but a gestalt representation of many observations in a flexible context. So the dog is able to generalize scent in a way that even if we were able to match the sensitivity in just pure detection, we wouldn't really be able to do the same kind of meaning making the dog does. And I think this figure from ICIT's paper in 2022, it's a review of volatile organic compounds from the human disease breath biomarker literature. Really illustrates well why it's so hard to generalize disease by molecules. So in the figure on the left is essentially showing, on both graphs, it's showing 4 different disease groups where you're just plotting the PCAs of all the VOCs that were found from GCMS across different studies. So this is a meta review. And you can see the blue circle is lung cancer, which is, you can see spread out all over the place. And the 3 other circles in between are 3 different diseases: diabetes, IBD, and liver disease. And you can see 2 problems. One, you can't really tell the 3 diseases in the big blue circle apart. So it's very hard to actually differentiate between diseases. And you see that the lung cancer, the blue circle is just all over the place. So it's also very hard to tell from a given set of VOCs or a given compound whether that's really a marker of the disease. And this goes back to a fundamental phenomena in olfaction and smell, which is different from vision and hearing, where you can't really parameterize as easily. With vision, you can say, okay, we have frequency or color intensities, RGB values. In audition as well, you can do frequencies. With olfaction, you can't really just break it down into 1, 2, or 3 different components. Instead, you have this combinatorial intractability. And there's this infamous example goes from this chemist Cell and Cell's triplets, where you essentially, in this first graph, you have these 3 odor molecules, which 2 of which are very similar in molecular structure, and then one of them is very dissimilar. And it turns out that the 2 similar molecules in structure actually lead to very different percepts. So one of them is vanilla and one of them is a citrus smell, whereas the 2 very dissimilar molecules in structure have very similar percepts. So they both smell floral. And a reason this happens is, in part, we think how the olfactory receptors are encoding the molecular space. And this is a joint embedding that we don't fully understand yet, which then creates this percept space, which is non-linearly mapped from the molecular space. And so if you use a machine that is purely seeing in the molecular space, you're not actually fully mapping on to the perceptual space where the real information and embeddings of what you care about might lie, such as the scent of cancer. And what we think at Diagnosis is the better way around the problem is what we're calling reinforcement learning by canine feedback, calling it RLCF, playing on RLHF. And here's Cheetah, one of our dogs, to really drive the point home. We thought this really captured her "I am correct" reaction. And what we think the solution to cells triplets and this computational intractability and the problem of looking at molecular space, instead of going molecular level up, let's go percept level down. Let's actually start with the percepts and have labels of the percepts and then work our way backwards to what the molecules could be. And how we think this fits with what we're doing right now is you have the dogs as perceptual labelers of an odor print's ground truth. So they know what the scent of cancer is better than any electronic nose or any instrument that we might have. And then what we can do with brain computer interfaces, machine learning, all the multimodal data that we can throw at the problem is actually create a perceptual transcript of the dog's own perceptual understanding, such that we have now this rich, multi-dimensional transcript of this label. Is there a question? Okay. And we think that's not the complete story. So I just tried to sketch out, and this is in some sense territory that we still are navigating. But you can have the dogs giving you the perceptual labels, you can have rich transcripts of these labels, but then you still have two missing pieces. One, you still have this combinatorial encoding by the olfactory receptors. And in some sense, you need to reverse engineer it if you really want that encoding magic that's going on. And then you also need to give the AI an actual nose. And these two pieces I'll just lightly touch upon towards the end about how we're thinking about it. So that's the big idea, RLCF. And now I'll just end in the next 10 minutes, which is talking about some concrete things that we're working on in our current R&D. Circa August 2025. So we have a experimental platform we're calling Sniff Space. Not the most imaginative name I know, but it's what we're now sticking with. We're thinking of it as an automated and gamified detection dog platform. So we have a reward delivery system. We manage samples. We give access to different ports. We have a suction system. Currently have both full body and face cameras to have accurate tracking of behavior. And then we have IR sensors giving us exact timestamps when sniff events happen. And we're further working now on automating further process of this. Essentially, the idea is the dog can walk in not knowing anything about scent work, and in a few weeks, without expert canine professional in the room, learn just how to play sniff space almost like an Xbox. So almost teach the dog how to work with sense, all the time giving us information and giving us that perceptual transcript for the sense in question, which we would do through what we're calling dog sense. So it's a wearable suit, currently has a channel EEG brain computer interface, fully wearable, non-invasive. We don't even shave the fur of our dogs because we don't shave people's heads. So why should we do it with dogs? We have some IMUs and we have a respiration sensor. And what we're doing, especially with the BCI, is essentially creating a fully custom headset to each dog. And it turned out this was actually quite a challenge. It's one of the challenges which have held people back from really thinking about wearable interfaces for dogs, because dog heads are really weird and kind of curvy and bumpy. And I mean, just very organic. And you don't have the same surfaces to work with that you do with humans. And so what we've done is essentially create a software pipeline where either with a 3D scanner or even a smartphone video camera, you can take a video or a scan and generate a fully realistic 3D model of that dog's head. And then we've generated a way in Blender to essentially sculpt a helmet for that dog, and then we 3D print that helmet so it has a fully custom conformable fit. And now what we're doing on top of that is to say, okay, we can also automate where the electrodes should go and in what montage based on the anatomy. And this is especially important because, again, with dogs and all the different breeds, you might want to think about. In our own team, we have 4 different breeds. You want to make sure that you're actually capturing from the same brain region across dogs. So we have MRI scans, which we've built then a canine neuro navigation algorithm. So you can have an MRI scan and then based on the brain regions of interest, automatically generate the montage in your 3D design that you can then 3D print. And that's also allowed us to do some source localization, which you have a lot of tools for in humans, but there is nothing really there for dogs. So we're essentially building it from scratch to then say, from that montage that we generate, what brain regions are we actually measuring from? And we've been experimenting around with a few different electrodes. We've finally settled on a semi-hybrid system where you have saline foam and then you connect it to a wet sintered electrode. We've been able to make it customizable so you can fit different electrodes into any helmet that you want to create. So this is a very old slide because it's taken us a while to really solve some of the dog ergonomics to be able to collect a lot of data. But from the EEG data that we have collected from a few months ago, we already were able to see that there were features that were different between target and non-target scent detection events. We think things like that we know from the human literature, like the P300 and other markers can also be found for odor detection. And the ultimate goal, and this is a slide from a former professor, Jack Gallant. I see Sheer is in the audience, so we both took the class together at Berkeley. The idea is essentially an encoding decoding model where, since we know what the stimulus is, we can, when we don't know what the stimulus is, map a response back to the stimulus. Now, of course, what we're doing with wearable non-invasive headsets can't really reach the level of fMRI scan. But what we think we can do is what has also been done already in the literature for humans as well as for non-odor tasks in dogs, which is develop a really good predictive model for when you have a subset of odors that the dog has sniffed in the past and you have those brain responses from those odors. Can you then predict a novel scent detection event and what odor the dog is detecting? So in human EEG, they've been able to do this with pretty high accuracy. This is a paper from 2022 in PNAS. And from the very small subset of EEG studies with dogs, they've also been able to notice differences when dogs are looking at, say, human and dog faces on a screen. And that's a far less salient feature than sniffing an odor that the dog thinks is the odor that gets them a treat. So we're pretty confident that with enough data, it's about just a matter of time before we can develop really good predictive models that can tell us what, or at least when a dog is sniffing, what it sniffed in the past. So another quest is to use GCMS as well to better understand the VOCs in question. And this starts getting to those two other pieces after you have the dogs labeling percepts and you start to get transcripts of percepts, which is, okay, what are the dogs actually sniffing? And we've just started now getting hits for what those compounds are that they are sniffing using GCMS with the caveat, of course. And I see we have a dog in the audience, which I'm very happy about. Once we know what they actually are sniffing, then we can start building that combinatorial encoding a bit better. Though we would also need to know the receptors, and this is something that I'm very excited to do potentially in the next 2 to 3 weeks on our own dog at home, actually, Lily, who needs to be spayed anyways. And while she'd be under anesthesia, there's a fully non-invasive procedure that was developed by this French team a couple of years back, essentially doing a COVID-like swab, but doing it where we know olfactory receptors are expressed in the dog's nose, which is not a trivial place to get, but you can get there. And what we're really interested in doing is if there's a really good non-invasive way to sample olfactory receptor expression, and we're able to do this in a reproducible way, then what happens when we sample from a cancer-sniffing dog and a non-cancer sniffing dog, especially if you control for things like the same litter and the same environment, then the difference in expression that you see should be as a result of learning the scent of cancer that actually gives you the receptors that the dog is expressing to learn the scent, which you could then start doing a process of predicting the shapes of those receptors and then looking at the ligands they bond and then actually finding the receptors, or sorry, the VOCs that those receptors bond to. And yeah, it's something that we're pursuing over a long time frame, but I personally find this really exciting because no one actually knows what receptors the dogs are expressing to sniff. We know what is in their genes, but we have no idea what actually matters. And the final piece is, of course, giving the future AI with the canine intelligence a nose. And a lot of people have built a lot of different sensors. We're working on this method using a quartz tuning fork, or QTFs, with another lab in India who've been working on this for the past decade or so. And we're excited about this because we think it some ways is analogous to what's happening with the olfactory receptors. And it's also very scalable and cheap to implement. And the idea, I won't get too into the weeds, but it's essentially a quartz tuning fork is this piezoelectric crystal. It's found very commonly in watches, and it vibrates at a known frequency. But when you dope it with certain nanomaterials, just with a coating, the interaction of the VOCs with that nanomaterial change that vibrational frequency, and it changes in a way that is consistent for that VOC nanomaterial interaction. So if you have a bunch of different quartz tuning forks, they cost like less than $1, and you coat them with these nanomaterials, and you can choose different ones, and then you run VOCs through them. You get this pattern which can actually tell you if there's this particular scent signature present. And what this lab has been able to do is prove this for diabetes, which is arguably an easier scent, but still quite complex. It's still in this buffet of VOCs in the human breath, and they were able to do this quite consistently. And what we're really interested in doing is, especially with the analogies with the receptors and what we learn from the dogs and those perceptual transcripts, we think this could be the final sensor that you apply RLCF on and have that AI with a nose that can finally maybe rival, or at least the dogs pass on the baton, is how I like to think of it, to artificial intelligence. And finally, all of this is stuff that we're really excited about from a scientific perspective, but we want to ground it in the clinical applications we care about, which is generating these correlation matrices with other kinds of clinical indicators, such as lab biomarkers, histopathology, radiomics, and even cell-free DNA, so that VOC detection by the dogs or an eNose has as much clinical context as possible. And the next step for us is to actually take the results from our first study into a prospective trial where we don't know the labels. And it's the ultimate test of clinical validation, and see if we can actually detect real world cases of undiagnosed and asymptomatic cancers from breath alone. So my parting bark before I open it up for questions would be that I think there's a very strong case that olfaction-based detection is the ideal first layer of screening. And it's for three main reasons. One, you have a system which is high sensitivity. So again, sensitivity is the ability to pick up a disease if it's present. And as we saw with our data as well as the literature, breath actually has biomarkers at a resolution that isn't really present in blood or really decipherable from other methods. So you can actually catch diseases. It's fully non-invasive, so you don't need to do any of the kind of procedures that require some kind of sitting in a scanner, a blood draw, anything else. And it's very low marginal cost, whether it's a dog, especially if it's a dog, or with a dog, it's a few seconds of their time. And thankfully, the dogs aren't yet charging us high salaries. So you can get away with each additional test not costing a lot. But even with the sensor, you can run multiple tests with a very low marginal cost. And all three of these mean that you have a system capable of constant passive monitoring, which allows you to provide the best possible risk stratification, who's more likely to have the disease, who's less likely, in a way that can really solve for Bayes' law. And all downstream diagnostics can be improved. So you don't really need to replace them. They still fulfill a purpose, but suddenly they can do a lot better since they're not dealing with that hard-based problem. 1 in 200 people have cancer at any given point. How do you actually test 200 people without a lot of false positives? You test people after you run them through an olfaction layer, let the nose filter all the people who don't need future diagnostics. And so that's what we really think is unlocked if you can really solve for the olfactory code. So that's me. Thanks so much for listening, and I'd be very happy to take any questions.
Thank you so much. And with that, we'll give a bark to open the floor for questions. I have a question, actually. So you mentioned that the dogs aren't really responding to specific molecules, but broader profiles of scent. I was wondering, are there scents, do the same scent profiles map across cancers? Like, can you identify which cancer a person has by which scent, or is it pretty much the same?
Yeah, it's a really interesting question. The dogs seem to be able to generalize very well. That's actually what got us on the track of multi-cancer. We thought it solves an important problem. If you do multi-cancer, you don't need to screen for each cancer. And so one test can be the first filter and then you screen for each cancer. But what we also saw, what my co-founder Itamal saw, who's been working with dogs for almost a decade now and has worked with disease detection dogs even before diagnosis, at his previous venture, he saw that they were training dogs to detect breast cancer, and they were then piloting out if the same dogs could also detect other cancers. I think actually the story went that there was another cancer by mistake in the set that they were running. I think it was brain cancer, and the dogs still indicated. And that was even though they didn't see any brain cancers before. And that's also what we saw throughout our process. The dogs are seeing a cancer for the first time. They've seen maybe 5, 6 other types, and they're able to still pick it out with pretty high sensitivity. So there's something in that signature of cancer, which is probably intrinsic to the biology of a cancer cell. It's producing all these metabolic byproducts that are unique to a cell that is multiplying on loop. But at the same time, what you see with the analytical tools, GCMS, is that you don't really have very neat overlaps of cancers. And we think even the dogs are probably seeing different shades of gray, but we just teach them like all of this is what we care about. So they don't bother telling us that there's different things that they're seeing. And so that's what we're interested both with the BCI ML. Can we actually tell each cancer apart if the dogs are seeing it that way? And also thinking about training specific dogs for specific cancers such that you can have a general physician dog and then a organ specialist dog, and then run a sample through both the generalist and the specialist to then get that information. But yeah, I think that's a long answer to saying we don't fully understand yet.
Wow, thank you so much. I think Micah has a question.
I have many, as always, but I'll just start with one. Did I understand correctly that the scent receptors, or the cells in the dog's nose that have the receptors on them, that expression pattern changes over time based on what they're exposed to? Or is it what they're trained to? Like, how's that work? I didn't know that expression pattern changed. I thought you just kind of were born with certain detection and that was it. I understand that's not actually the case.
Yeah, so I'll clarify that we don't know with dogs. So this graph on the left is from mice, but it's quite well reproduced in mice so you are born with a set of olfactory receptors in your neurons, but what can change is which receptors and which neurons. So each neuron carries only one specific receptor, and that's actually quite flexible. So the olfactory receptors and olfactory neurons and the olfactory region in general is very neuroplastic. It's one of the regions you see neuroplasticity in adults. And what the mice studies in this graph shows is that if you expose mice to certain odors while they're young, while they're growing up, even from the same litter group, depending on the odor, you see a differential expression in certain olfactory receptors. So that's kind of what's being shown in these 3 bar graphs. It's the 3 different groups of mice. And there's a lot of other things this paper did. But yeah, in general, it seems like an odor imprinted with a certain salience causes an expression of receptors which are specific to that odor in some form. And so if it's there in mice, and I think there's some literature in humans as well, then it's probably also present in dogs. But we don't really know. No one's done actually an expression before or after odor stimulation with dogs. But it seems like it should be the case.
Great. I think Abdul Rahman had a question in the chat. I can read it for you, or you can unmute and ask it yourself, whichever you prefer. Okay, I'll go ahead and read it. It says, how do they represent many observations in flexible context in the brain?
Yeah, again, I think probably skip the long answer and just say, I don't think we know. It's something, I mean, something interesting about the dogs and kind of the vision analogy that I used a couple of times is that dogs seem unique in having a dedicated tract from the olfactory bulb or the olfactory region. So once it goes from the bulb to the olfactory cortex to then the occipital lobe, which is in the visual cortex in humans and most mammals. Instead, with humans, we don't have this tract of connecting the olfactory bulb to the visual cortex, but dogs do. I don't know if that's the answer to the question of how they are generalizing, but it seems that there's something that they're able to do with context and multiple exposures and this incredible sensitivity, that the raw power that they have, and then just something that's going on in their brains that, yeah, that we would really like to know, but I don't think I have a good answer yet.
All right. Victoria, would you like to go?
Yeah, I have two questions. One, have you tried any of the G-Tech animal recording EEG? I know they also have some options they've been showing on horses or cats and dogs.
Yeah, I've come across it. We haven't tried it. I think from what I understood, it's a cap-based, but it's not custom to each dog. And we've also tried cap-based, but we found that especially when there's movement, which I think not a lot of people are trying to do wearable EEG for dogs or animals. Most of the time, you're probably doing it when the animal is still. But when there's movement, just again, the ergonomics of a dog's head are just difficult to design for. So when we've used kind of loose spandex type helmets, there's still a lot of movement off the electrodes. It's hard to kind of anchor it down. And that's why we have a more like TPU, soft, rigid structure for the helmets to keep those electrodes always on the scalp. Be curious to know, yeah, what they're doing. But my sense is building a custom helmet for each dog and then having this 3D printed model gives you a bit more room to do wearable stuff than what you would otherwise be able to do.
Yeah, that's super interesting. I've done a little EEG in people myself, and it's always the dream to do it in more active situations, but like the motion is so bad even when they're sitting still. You have to tell them to sit very still. So it's kind of an interesting idea. Maybe the solution will be doing these kinds of scans for whoever the subject is. My other question was how, kind of more broadly, I guess, as a founder, how do you get people to take this project seriously and strike the right balance of being accessible and also trying to come up with a business proposition?
Yeah. Just in the last comment on the first question, though, I think we have wondered about, and probably there are companies, maybe someone in this call would know of generating custom 3D printed headsets for humans. Because, yeah, as you said, I think it's also a problem with humans and asking them to stay still, and asking our dogs or any dog to stay still is even harder because they don't necessarily cooperate as nicely. So it's, yeah, it's something that we wondered about whether this is already happening with humans. Maybe it is. Yeah, with the second question, I think initially a lot of people, I mean, we had a hard time. It just sounds like a really weird, fun, but also not really serious idea. And the juxtaposition of dogs, cute, and then cancer detecting high stakes was like something people took time to like get used to. But I think, like we were just talking at the start of the call, Lydia and I, we already depend on dogs to detect explosives in these high life death situations before boarding a flight, for instance. And once you have some of these framings, it's a lot easier to digest, I think, what we're doing. And so now we no longer, I think, have much of a challenge convincing people this is a real idea, and it helps that we have really good data. We're publishing that data, presenting actively the science. And I think another part, the last part of the question was balancing, I think, the science and kind of the business aspects. And I think it just helps a lot to have a great co-founder who is thinking a lot about the other parts that like I can't think about just because of bandwidth. And then also just a great team to support you, both humans and dogs.
All right. Thank you, Akash. Nice talk. We were mentioning 2 slides ago or 2 questions ago that there's a strong track from the olfactory bulb to the visual cortex, hinted at that being some generalized kind of sense. And I was wondering if you also were implying that dogs see smell, if that's like a fair analogy to me.
Yeah, I think that's what I was hinting at. I think there's, I mean, we often in diagnosis also resort to visual metaphors because, again, humans blinded by vision. But there's a sense that we generalize pictures really well. We can see, like, yeah, just the ability for us, it's still, I think, we're just orders of magnitude better than LLMs or current architectures at being able to see very few samples of like a certain picture or something and then be able to like know what the next one is or means. And I think just because we have a hard time really thinking concretely about smell, but it's probably no longer a metaphor if there's this track that goes from the olfactory bulb to the visual cortex. And there's probably something really interesting about extending the metaphor quite literally about dogs seeing smell and this idea of like synesthesia almost happening. But yeah, again, it's just so hard to think about, I think, smell and describe it with language. It just kind of breaks down.
Thanks so much. Micah, would you like to go?
Sure. Rather than using the tuning forks, would it be possible to culture cells that have the same olfactory receptors as the dog cells and then have those cells do something detectable like express GFP or something when the receptor is bound? And if that's reasonable, what do you see as the advantage of using like a mechanical device like this versus some sort of biological device?
Yeah, no, it's really an interesting question. There's at least one company working on this, trying to cultivate olfactory neurons. They're called Conico. The idea is, like you said, have the neurons live in some kind of vat and then you keep them alive and connect them to a bunch of electronics. I think keeping cells alive is hard. So that's just hardware, maybe software, not software, just yeah, wetware limitation. Maybe the technology will get there where it's less hard, and then you have other problems. I think another response to that would be why not just grow the receptors? Forget about the cells, just have the receptors live. I think we're starting to have technology that can do that. I was just talking to someone who's thinking about that, and I think that could be really interesting. And you're right, then it wouldn't really make a lot of sense to use the forks. It'd be much nicer to use the receptors. I'm personally really interested if like that research gets mature enough where you can actually start doing that. I think with the forks, what we really like is just how kind of foolproof they are. It's like super cheap. The synthesis of the nanomaterial and the coating isn't very complex. So you can have a lot of these devices and sensors and you can collect a lot of data and already start making sense of it. And then maybe you can just replace the sensor once you have better like biosensors. But yeah, it's a good question. Why not just do it with biology all the way through?
Thanks. What is the false positive rate of dogs detecting cancer? You talked about sensitivity, but I'm curious about the inverse.
Yeah, the sensitivity is something that I highlighted, the specificity. So here's the ROC curve and kind of different values across the decision threshold. We've been able to get a 90% plus specificity and sensitivity in this test set. You can have as high as like 95% specificity and drop your sensitivity or vice versa. And this is mostly completely with the dogs. So we've had some like Bayesian ensemble with historical performance priors, some of the IR data. We haven't really integrated the vision data or the EEG yet. That's still a work in progress. So the dogs can be pretty sensitive or pretty specific. not as specific as some of the genomic tests, though. And that is something that I think they're just going to be better at. The liquid biopsies will be able to tell you with a much higher percentage or accuracy whether someone indeed has cancer if the test says you have cancer. And that's why the focus on sensitivity is complementary to the focus on screening and being the first layer. You want a sensitive screen, you don't necessarily need a specific screen if you're the first layer.
Okay, thanks. Last one. It seems easier just to breed one dog rather than building out a whole bunch of specialized hardware that conforms to every possible dog breed. What is the advantage to going through all this effort to make this headset and whatnot that works with all dogs rather than just breeding a dog with a specific head shape?
Yeah, so I mean, interestingly enough, even in the same breed, so I have an image on the left here. It's a composite of 6 of our beagles, where the yellow is the areas of their head which differ significantly, and the blue is when they are pretty similar. And so 6 beagles as well turn out to have like pretty different head shapes. Some beagle helmets fit the others, they have similar heads, and then some don't. So I think you'd still benefit from making potentially custom or at least fine-tuned helmets if you really care about getting as good data as possible. I think the other response would be that having a wider pool of dogs you can work with. One of our dogs is adopted from a shelter. She's an indie lab mix, and she's actually one of our best dogs. And just having a much wider pool and saying that you can work with dogs from shelters, even in a future model, dogs at home, instead of going to daycare, they go to like a sniff gym where they put in some olfactory reps, but also like detect disease in the process, I think was something that we really like the idea of in terms of scaling it, and also the other use cases of the helmets. That's why we put in the effort to just get a conformable head. You could also do what the Koreans did, which is just clone the same dog and then have a team of clones in their airports, which is what South Korea currently has. It's 8 members of the same dog. So they went that route, which I guess is each method has its pros and cons.
I have too many questions, sorry. One, do you happen to know if those Korean dogs all have the same head shape, or do they still have diverse head shape despite being clones?
Yeah, I don't know. We'll have to ask them, I guess, to measure
That's
the
fine.
head. I have been curious about, it's just once you have clones, kind of interesting. Even the olfactory receptor question would be very interesting, but
Yeah.
we haven't reached out to them yet.
Okay, and last one, I swear. Do you find that there are different, or do you know if there are different olfactory receptors by dog or by breed, or is it kind of universal? They all have the same set?
Yeah, so we know that they have differences in genomes. So a German Shepherd and a Beagle can have 40, 50 different olfactory receptors out of the superset of 900 that they have. But the expression we don't. And that's, I think, actually where probably differences in detection ability from a receptor level come from, is that different breeds are expressing different genes or different receptors differently. And yeah, that expression we just don't know. It's a black box.
All right. Well, that brings us pretty much to the hour. So thank you, Akash, for the incredible talk. It's been totally fascinating. And we're really excited to see what you all achieve next. And thank you, everyone, for joining. Thank you for your time. It's been a total pleasure to sniff and bark with you for this past hour. And hope to see you next month.